definitely disagree with, but I'll just give it to you ad arguendo. Then
Well, I don't want you to just give it to me. Let me just ask you, because I'm thinking, by its very existence, a nucleotide cannot be neutral. So I don't want you to just agree with me. And why I believe that is because nucleotides, they do take up space. They affect spacing between other nucleotide sites, and they even affect regional nucleotide composition. What else? DNA folding, nucleosome building. So that means even if a nucleotide contains absolutely no information, it still cannot be neutral because it's still going to slow replication and even waste energy. So I'm just saying
you're
it seems
confusing.
evident that there are really no neutral mutations. Like, would you
You're
agree with that, or would you still have to concede that?
Yeah, so you're confusing the fact that there are regions where spacing is relevant for the, and you're taking that and you're running that and extrapolating it out to assume that all spacing is relevant, and that is demonstrably false.
Well, I think it also goes back to obviously the non-coding DNA, because you guys would say that the non-coding DNA absorbs the mutations, right, making them neutral. I know we talked about that
And also,
extensively before, but I want you to comment that. But what I'm saying is, if this non-coding DNA, let's just say it does help during embryonic development, and it can be part of many diseases, which obviously they're finding out now. And just because scientists cannot see any function per se in some of these non-coding DNA regions, that doesn't mean they're non-functional. So that would make the majority of mutations near neutral versus, say, neutral, right? Go ahead.
right. So what you just said is that if this is true, then something else is true. And the problem is that not only is there not evidence that it is true, there is strong evidence that it's actually false.
Well, if we look at, and I want to give an accurate representation of what ENCODE found, and tell me if I'm representing it right, okay? So ENCODE would have estimated that 80% of the genome is functional, but in the sense, okay, and I get what you're saying, that it has specific biochemical activity, right? So I think it was an ENCODE researcher that said, it's like opening a wiring closet and seeing a hairball of wires. We try to unravel this hairball and make it interpretable. So he's saying that obviously a lot of this function, it's believed to be regulatory, right? So the DNA is telling the genes when to produce the product.
No, that's not right. I'm sorry, that's not right.
Okay, go ahead.
It's not believed to be necessarily regulatory. When they say that it has firstly, there are two errors there, which is it isn't that the single nucleotide is involved in specific biochemical activity necessarily. Instead, what they're saying is that these nucleotides are within some distance of something that is involved in a specific biochemical activity. That's the first distinction, right?
And would that fall back to your spurious transcription?
No, this isn't. We're not even there yet, right? So basically, what they're saying is, let's say we know that some transcription factor has a certain consensus motif, right? So I'll just make one up. There's a transcription factor that binds to, and I'm going to type it in the side so I don't forget it. It binds to GCA TGA. That is the consensus sequence for this transcription factor. So what ENCODE would say is that every time you find GCA TGA in the genome, you go 1 kilobase in front of it and through 1 kilobase behind it, and you say that 2 kilobase region, all of it, is involved in a specific biochemical activity. Now, the fact that there's a binding motif doesn't mean that you ever get transcription from there. It just means that it matches the consensus region of a binding motif, right?
But how come we find all independent functions, the ones we're looking at, they seem to be in that 80% area?
Well, because the 80% area includes everything almost, except for the only thing that it doesn't include is that other 20%.
Okay,
right?
so
So what are the odds that most of the stuff is included in 80%? Well, it's pretty good.
but if every month we're seeing more, okay, for one, I want to go back. So obviously we agree that ENCODE did define function as specific biochemical activity, right? I'm not
That is
misrepresenting that.
that
But
is what they defined it as, but I don't think that we
okay,
have sufficiently explained what they mean for the context of this conversation.
but did ENCODE find the majority of DNA is transcribed into RNA? You know, copying the
Um,
DNA into RNA? Because
they
it
found
was thought to be exclusive to parts of the DNA that coded for proteins.
that's
Now we're
false.
seeing that activity in the non-protein coding
No,
regions.
I'm sorry. Spurious. Now we are to spurious
Yeah,
transcription.
so now it goes back
Spurious
to,
transcription was a known phenomenon for decades before ENCODE.
now
Now,
would you say,
you could say that they didn't know the entire extent of it, but they knew about it.
but what I'm saying is, would you be disagreeing then with a lot of the ENCODE researchers? Because I know one said, based on the findings, he said, we are the most complex things we know about. It's not surprising that the manual is huge. I think it's going to take this century to fill in all the details. So what he's implying is that it would take a century to understand 100% of the DNA language. So it seems like a lot of the quotes and writings of these ENCODE researchers, it seems like they're doing their science, without that evolutionary bias, and they seem to be suggesting that our genome is chock full of functional information. Like, would you disagree with that quote, or do you think that that's just a misrepresentation of what he's
You're
saying?
asking me to speak on a quote from someone I don't know in a context that I'm not familiar with, right?
Okay, well,
Like
okay, if you're saying spurious transcription then, but we're seeing that much of the 80% say was found to be controlling also the expression
that
levels of,
isn't what was found. I'm
no,
sorry, you're simply
we know
wrong.
but we do know that non-protein coding DNA is telling the protein coding genes when to produce their product, aka the protein, and when
Okay.
not to. So
So again,
that is a clear trajectory.
here's a fundamental error in your thinking, right? Which is that just because some things within category A do thing X,
Okay, but
it
you do
doesn't mean
agree
that
with
all
what I just said,
things in
though.
category A do thing X,
But it seems
right?
like you never agree with the specific expressive and regulatory roles in these specific functions I'm describing. You do
Well,
agree
yeah.
that those are found in those non-protein coding
Well,
regions,
look.
though, right?
I'll give you an example. There is a gene XIST, I believe, is the symbol for it, which is involved in sex differentiation, if I'm remembering all of this correctly. And that is a long non-coding RNA. Is very important to proper development, but it's not a protein. Okay, now the fact that that exists doesn't mean that all long non-coding RNAs have very important functions. It means that it is possible for a long non-coding RNA to be functional. It doesn't mean that just because you found a long non-coding RNA that that thing does actually have a function. Now, the same applies to all of the other examples that you might point out. The fact that it is possible for something of a certain description to have a function doesn't mean that all things of that description have a function.
Okay, so when you look at things like, regulating DNA replication, regulating transcription, marking sites for program, rearrangements of genetic material, and obviously I can go on and on, controlling RNA processing, editing, splicing, modulates translation process. Obviously, it even regulates embryological development in some sequences. All of this is found in the non-protein coding region, and the fact that we're finding more and more.
All right, so I'd like to ask you right now, do you even understand what my point was?
No, I understand what is the things that I'm listing. You're saying that it's in those regions, but based on that spurious transcription, it doesn't mean that it's.
No, that's not what my point is.
You're agreeing with the functions that I'm
Let me
saying,
put it this
but
way.
you're saying it's
Let me
such
put
a
it this
small
way.
percentage of
Let me
it
put it this
that
way.
it means nothing.
Hold on. Let me put it this way. let's say you have the entire alphabet of letters, okay?
Yeah.
And I tell you that the letter A has a function, okay?
Okay. Yeah.
Does E have a function?
So you're saying the
You
letter
don't
A has a
know. You
no,
don't
we
know if
don't
E
know.
has a function.
We don't know. I admit that
It's
we
in the
don't
same.
know a
It's in
lot
the
of
same
these non.
category. It's in the same category as the thing you know has a function, but you don't know that it has a function yet, right?
Are you
So
familiar? Are you familiar with the article called "Immortality of Television Sets"? Because it was based on evolutionists who actually criticized ENCODE for not using evolution as a starting point. So it seems like there's this huge bias based on everything that has been discovered, because it's showing that, you know, these sequences aren't idle. Okay, so
no, but
obviously a lot of, just wait. So if a lot of this genome is transcribed, okay, and you know, it, if it is just spurious transcription, I'm just saying a lot of energy for the cell to do something like that that doesn't actually have function. That's
so now let's move to another point, and
okay.
it's related. It's a very closely related point. Imagine, if you will, that you create a 10 kilobase sequence that is completely random, and you take that and you insert it into some simple eukaryotic cells, into some yeast cells, right?
Yeah.
And then you get it incorporated into the genome, and you grow up these yeast, so you get a bunch of them, and then you do deep transcriptome sequencing on these things. So you have this big sequence, 10 kilobase, right, of completely random order. Okay. Now, if you were to find that of that 10 kilobase, that 60% was transcribed, it on at least had at least one read from at least one cell in the entire experiment, that at least that 60% of that completely random sequence was transcribed. And then you saw that from the rest of the genome of the yeast, that 80% is transcribed. What does that tell you? Because it should tell you something very specific.
No, it actually would tell me that, based on the fact that we know very little about the genome and we're discovering more and more every single day, that this does suggest and is a strong trajectory for genome-wide function. Because all I'm seeing from you is evolutionary bias.
Okay,
I'm
so
also
you've
seeing a
missed
rescue
the
device,
obvious.
and I'm
You have
seeing
missed the extremely obvious statistical implication there, which is that 60%
I know what
is noise.
I know
Right.
what I know that noise is the rescue device, but if you actually look
It's
at
not
one
a
of the first,
rescue device.
just
It
what
is
I just want to say something.
demonstrable. You
If you
can
look at one of the first,
demonstrate it.
if you look at one of the first junk sequences that we actually found doing something, it had to do with, and you're probably more than aware of this, had to do with males and females, because we know males have one X chromosome, females have two X chromosomes. Now, a double
Yeah,
dose.
you're talking about the thing I was just talking about.
Yeah, and what I'm saying is they discovered that because half the genes on the X chromosome are deactivated or activated, while the other half will deactivate. So on the woman, so that actual
It's
important
not half the
function.
genes, half the chromosomes. It's
Well,
far body formation. It's random X inactivation.
well, it's half the genes on of the X chromosome will deactivate,
No,
and the other
no, no,
half
no.
will
That's no,
deactivate
that's
on
no,
the
that's
other
wrong.
one.
That's wrong. I'm
No,
sorry.
it's
That's
what.
wrong.
Let me explain myself without you actually interrupting. Okay, so what they found, because we know a double dose of the genes on the X chromosome would be bad. Okay, now half of the genes on the X chromosomes are deactivated, and the other half will deactivate on the other one. So anything a female needs,
Okay, so you
a
wanted me
double
to
dose
tell
of
you
let
why what you're
activate and
saying
operate
is
on both
wrong?
chromosomes.
Okay.
How is that wrong?
You know, because what's happening is that one of the sister chromatids, right, is inactivated the entire length of it. Okay, all the way up and down, it gets inactivated, right? So you have, right, one copy of each gene is inactivated, but it's always the copy from the same side. Okay, that means that that in this cell for this chromosome, either the one you got that in this cell for the X chromosome, either the copy of X that you got from your mother or the copy of X that you got from your father is entirely inactivated, right? One or the other.
I'm
in
talking
this
about.
cell,
I'm talking about
up and down.
I'm talking about the woman though. A double dose of an X chromosome is. I'm talking about the actual function that they found in these. Because I think you're missing the point. What they found out is that a portion of the junk sequences was actually calling all the shots and was coordinating these activities that we're talking about. So
Yes,
all the
XIST.
Yeah, so all the junk sequence, obviously these sequences that aren't coding for proteins actually tell all the other
tells
sequences
you that one,
to do it, how to do it, how much to do it.
No,
I mean, so many overlapping roles there.
I'm sorry, you cannot make that assumption on the basis of that evidence. You can say from that it is possible. It is possible that a non-coding RNA can have a function that was already known prior to the discovery of the function of XIST. They already knew that. And so guess what? They were kind of looking for something like that because they knew those things could be functional. But the fact that things can be functional doesn't mean that everything of that same description is functional.
Exactly why I say the word trajectory every time. Let's just say, for now, for sake of argument, which I don't agree, let's just say it is 80% junk or whatever number you want to throw on it. Can you give me a step-by-step in the DNA on a genomic level of the very, very busy, I'm sure we'd agree with that, embryonic developmental stage? Not the growth factor, but what the DNA actually does, step by step, building the embryo, aka all the construction tools, everything that goes into that. Can you give me that?
Can I give you that like right now in this hangout? No.
Now, a lot of that is unknown. And what we're saying and predicting is that a lot of that process is taking place in those non-protein coding regions. A lot of those developmental stages.
I encourage you to demonstrate that claim. It is not incumbent on me to demonstrate that claim of yours.
How I'm demonstrating is based on the 80% of the non-coding DNA that's transcribed into RNA that's suggesting function, based on everything that I've showed here. That's like what it is. It's a trajectory for this genome-wide functionality because we know very little about these embryo.
It's absolutely not
stages in the embryo. Because I urge you to go look it up. They know very, very little about that step-by-step process.
I encourage you to go look it up because they actually do know a lot about it.
It's simple, Crispr. We don't know what DNA does in development per se, and based on the fact that there's a strong trajectory here, we're saying, based on pre-existing heterozygosity, that it makes testable and falsifiable predictions. This is the prediction based on
why haven't you tested it?
Well, the thing is, test it. Okay, scientists perform knockout experiments to determine the function of DNA, obviously in non-human species. We know how that's done. In this way, obviously, the functions of sections of DNA can be determined, but comprehensive, we agreed to this last time, knockout experiments for a mammal have yet to be performed. So it is
just wrong about that.
So I'm saying it is premature for us to say that it's fully functional, but it's also premature for you to make the claim that the majority of non-coding DNA is non-functional. Are you saying that you've done these gene knockout tests on humans?
I haven't done them on humans, no.
And that's the point. So we need to know what's going on in the DNA on a genomic level during the developmental stages.
They've done them on mice, though.
But the thing is, have they done?
You understand that they have done large-scale genomic screens on mice where you would end up with 2 to 3 to 4 to 20 or however much megabase knocked out of a chromosome. Okay, and these mice, they'd keep breeding, they'd keep living. Maybe you'd see some minorly altered phenotype here or there from 20 megabase
Yeah, but all we know is the growth factor of embryonic development. So, and that goes for mice as well.
Well,
They
obviously,
haven't looked
if
at
it's
that in mice. So those construction tools
what?
have yet to be
Are
discovered
you
in
kidding me
mice as
right
well.
now? Look, maybe you didn't hear what I just said. What I just said is that mice with huge sections of their genome knocked out have baby mice that develop through all of the embryonic development, also with the huge stretch of the genome knocked out, and they're fine.
Now, remember when I said a lot of these gene sequences that are involved in the embryonic development are silenced afterwards? If that knocked out region has been turned off after the mice embryonic stages, then that is very telling and very much
I'm sorry,
expected.
I think you still didn't hear what I said. What I said was you have let me go into more detail. You have a mama mouse, right, and a dada mouse, and both of them have the same big ass region of the genome knocked out, and
Yes.
they have a little baby mouse. And guess what? Both copies of the DNA it gets from the mom and the dad are missing this big old section of genome. So it's never there in the first place in the fertilized egg.
How
So
do
it
you,
can't be present for any of embryonic development because it was never there in the first place. So
which
you can't
section,
activate it.
yeah, how much of this section of the genome that was knocked out, what makes you say that that is that specific section that is involved in the embryonic development?
Well, let's see. If they do this repeatedly, which they have, and they knock out lots of different sections, which they have, they can cover a lot of the genome. And guess what? Most of it doesn't do much.
So would you say that you look at a lot of this in an unbiased point of view, or would you say that you look at it in more of an evolutionary bias point of view? Like, do you
I,
suspect any type of future discoveries? Like, would you put that in print and make that prediction that what we're going to find uh, is going to be just more non-functional coding regions in these areas, or do you suspect that we're going to find more various forms of functions? And secondly, in regards to the mice, how much percent of the mice did they actually knock out? Because I want to hear the numbers and obviously not just stories in order that I can accurately address what you're saying. Go ahead.
all right. So, firstly, let me see. Um, yeah, here's the one I want. Um, yeah, I think this is the one. Ian, these are the ENUs I want, I think. Early methods, sure. ENU. Yeah. In ethyl in nitrosourea, which used to create rats with by mutating somatic cells and all this other stuff. Yeah, so this thing would produce pretty big changes. I'm trying to see if I can find a big meta-analysis. I'd have to get back to you with the meta-analysis of it because I know
Okay,
I've got some. I've got.
okay, no, hey, that's fine. You can get back to me. But what I want to say is if we actually look at the history of genetics, okay, and I'm sure you know about it, very well, even back in the 40s, what was it known? One gene, one protein, one function. That means many things have been overturned. RNA, you can even look in textbooks. A lot of professors and scientists back then used to say that it's very much underappreciated. Okay, so a lot of this junk DNA is what I'm saying is decades ago biology, because you can see massive literature now showing unexpected functions of RNA. Right, so we're looking at a highly compressed DNA code beyond the best computer code. Because you'd agree, we write books, but we don't write books that can read forward and backwards. Even if you look at something like protein moonlighting, right? What did they see or what did they think of in the 40s? One gene, one protein, one function. Protein moonlighting suggests that these proteins are doing numerous things you wouldn't even anticipate. It's the main tool, or one of the main tools, I should say, for for building the body, and yet it's a multifunctional tool. So what I'm saying is, the cell uses possibly, these redundant elements to slow and speed the process. I mean, based on this highly compressed DNA code, I would say that the trajectory is strong for genome-wide function, but obviously, we'll have to see what the future makes out for that, and obviously agree to disagree on that. But that's where we go to